Students, we are discussing the chapter Biomolecules, and today we'll discuss nucleic acids. So what are nucleic acids? Nucleic acids are macromolecules found in the acid insoluble fraction of the cell. So we know that nucleic acids, these are organic compounds, as these are derived from living bodies, or these are found in the living bodies. These are again macromolecules, and these are found in acid insoluble fraction of the cell. Nucleic acids are polymers of nucleotides. Now, what are nucleotide, or the structure of nucleotide? I have already explained in our previous class. So these nucleotides are the monomers forming nucleic acids. Now in nucleic acids, as these are the polymer of nucleotides, so the adjacent nucleotides are held together by a particular bond which is known as phosphodiester bond. So nucleic acid bur hoise macromolecule aru ihotok pae acid insoluble fraction, cellor acid insoluble fraction not pae. Nucleic acid bilak hoise polymer aru iar monomer tu hoise nucleotide. So ei nucleic acid polymer bilakot duta usora usori koi thoka nucleotide eta bondedi particular bondedi log lagi thake, and this bond is known as phosphodiester bond. Okay, so here these two adjacent nucleotides which are held together by a phosphodiester bond. Now coming to the types of nucleic acids, that also we had already discussed in our last class. So the nucleic acids are of two types. One is DNA and RNA. The full form of DNA is deoxyribonucleic acid, and RNA, it is ribonucleic acid. So here we will discuss one by one. So what are deoxyribonucleic acid? So Watson and Crick have proposed a double helical model of DNA. So that model we will discuss now. So Watson and Crick model of DNA. Point number 1, DNA molecule consists of two polypeptide chains or strands. So DNA molecules, these are double helical, and it contains, or it consists of two polypeptide chains. Like this I am showing here, these two polypeptide chains. Now next point, these chains form a double helix like spiral staircase. So these two are the polypeptide chains, so they form a double helix. Like this. Okay, so they form a spiral staircase like structure. Now the sugar phosphate units form the backbone and the base form the center. So we know that each monomer unit of nucleotide consists of nitrogenous base plus pentose sugar plus phosphate. This is the composition of a nucleotide. So here sugar and phosphate, these two units form the backbone of the nucleic acid, and the base form the center. At its central point, nitrogenous bases are present, like suppose adenine, thymine, guanine, cytosine. How? I'll show you. Repeating again, DNA molecule consists of 2 polypeptide chains. DNA molecule দুটা polypeptide chain থাকে। এই চেইন দুডালে ডাবল হেলিকেল স্ট্রাকচাৰ বা স্পাইৰেলি পাকখোৱা স্ট্রাকচাৰ এটা ফৰ্ম কৰে এনেকে। আমি জানো যে ডিএনএ হৈছে এটা নিউক্লিক এসিড আৰু দিস ইজ এ পলিমাৰ, এটো এটা পলিমাৰ হয়। সো ইয়াতে মনোমাৰটো হৈছে নিউক্লিওটাইড, and nucleotide is composed of nitrogenous base, pentose sugar and phosphate. So here this pentose sugar and phosphate unit, pentose sugar and phosphate log lagi pelai, they form the backbone of this structure, and nitrogenous base they form the center. So nitrogenous base bilak bhitorot thakibo aru baki side ot pentose sugar aru phosphate thakibo. Now both the strands are joined together by weak hydrogen bonds. So here these two strands are joined together by weak hydrogen bonds. So these hydrogen bonds are the linking bond which connect two polypeptide strands. Point number 5, the two strands are anti-parallel if one runs in 3' to 5' direction. So this is known as prime. The other is in 5 to 3' direction. So both the strands are anti-parallel, 3 to 5. If one is 3 to 5, these are some measurements. Another is 5 to 3'. Okay, so here also as they form the coil structure 3 to 5 and 5 to 3'. Okay, so both the strands are anti-parallel. These two strands are connected by weak hydrogen bonds. Now the width of the DNA molecule is 20 angstrom. The helix takes a complete turn after 34 angstrom. So one complete turn. So I am showing this is the complete width of a DNA molecule, 20 angstrom. Now the measurement of a complete spiral or complete turn till this much, this is 34 angstrom. Okay, there are 10 base pairs in a complete turn. So one complete turn consists of 10 base pairs like this: 1 2 3 4 5 6 7 8 9 10. So one complete turn, its measurement is 34 angstrom and it consists of 10 base pairs. This is one pair, another, third, fourth, fifth, sixth, seventh, eighth, ninth, 10th. So it consists of 10 base pairs, and the difference between two base pairs, it is 4 angstrom. So eta complete turnot thake 34 angstrom. Complete turn to 10 base pair thake aru দুটা base pair major distance to hoise 3.4 Armstrong. Is it clear? Now, one chain can have any sequence of bases, but the other has to be complementary. If there is a purine base on one helix, it has to be pyrimidine on the other. This is called base pairing. It is very specific, and the only pattern followed is adenine and thymine, guanine and cytosine in case of DNA. So adenine and thymine, these two are linked with 2 hydrogen bonds, and guanine and cytosine, these two are linked with 3 hydrogen bonds. So base pairing, it is very important. Here I am showing, look, if one strand contains adenine base, then in the other strand it must be thymine, and these two are linked by 2 hydrogen bonds. If the first strand contains guanine, then in its opposite position in the other strand it must be cytosine. Okay, and these two are linked by 3 hydrogen bonds. Then again I am showing, if it is thymine, then on the other side it should be adenine. If it is cytosine, then again guanine. So purine always pairs with pyrimidine. So purine always form a pair with pyrimidine. So purine ki ase amar? Adenine and guanine, cytosine and thymine and cytosine, and this is the condition. Adenine hodai thymine logot log lagibo aru guanine hodai base pair koribo cytosine logot. Opposite hobo pare, but this is the formula. And 2 hydrogen bonds are present. 2 hydrogen bond adenine aru thymine er majot thakibo, guanine aru cytosine er majot 3 hydrogen bond thake. And last point, the nucleotides in a helix are joined together by phosphodiester bonds. I have mentioned already. So in a helix, the nucleotides are joined together by phosphodiester bonds. So 2 nucleotides are joined together by phosphodiester bond. So this form of DNA, this structure, this is called bDNA. Okay. Look, here I am showing the structure of a DNA molecule. So these are the central base pairs, and at its backbone this is phosphate and sugar, then nitrogenous bases. So I am showing the complete structure here. This is the sugar, this is phosphate, this is nitrogenous base of one nucleotide. Okay, then again in the opposite strand, nitrogenous base, sugar, phosphate moiety, another nucleotide. So here the connection is between, so the weak hydrogen bond or 2 weak hydrogen bond connects the 2 nucleotide through the nitrogenous bases. Here nitrogenous base is adenine of the first strand, then on its opposite strand it is nitrogenous base is thymine. So these are linked together by 2 weak hydrogen bonds. So this is one complete nucleotide: phosphate, sugar, nitrogenous base. Phosphate, sugar, nitrogenous base. Is it clear? And the nucleotides in a helix are joined together by a phosphodiester bond. So this is the Watson and Crick model of DNA double helix. So this is very important for your exam purpose. Go through it. If there is any confusion, then let me know. Okay? Thank you. Now, the next nucleic acid is ribonucleic acid or RNA. So RNA is a single-stranded polymer of ribonucleotides. So RNA, it is single-stranded. So DNA to ki asile double-stranded or double helical, but RNA etai strand thake, and it is a polymer of ribonucleotide. DNA to polymer asile deoxyribonucleotide. So here in RNA, the pentose sugar is ribose, but in DNA, the pentose sugar is deoxyribose. The nitrogen bases are adenine, guanine, cytosine and uracil. So these four nitrogenous bases are found in RNA, but in DNA, in place of uracil, thymine is present. That means the nitrogenous bases found in DNA are adenine, guanine, cytosine and thymine. Now coming to the types of RNA. So there are three types of RNA. One is messenger RNA or mRNA. M should be written in small letter. Transfer RNA or tRNA, ribosomal RNA or rRNA. So this is all about RNA or ribonucleic acid. Okay. Next is nature of bond linking monomers in a polymer. So monomer বিলাক কেনেকে লগ লাগি কেনেকুৱা ধৰণৰ বন্ডেৰে লগ লাগি এটা পলিমাৰ ফৰ্ম কৰে সেইটো আমি এতিয়া ডিসকাস কৰিম so there are various types of bonds linking the monomers to form a polymer so to form a polymer monomers must be linked with a particular type of bond some of the important ones are described below first is peptide bond amino acids are linked by a peptide bond, isn't it? We have already discussed. So, two amino acids are connected or linked together with the help of a peptide bond. Now, how a peptide bond is formed, we have already discussed. Next is glycosidic bond. The monosaccharide molecules are joined together by a glycosidic bond. So in a polysaccharide, each monosaccharide or two monosaccharide units are joined together by a glycosidic bond. So this glycosidic bond is also formed by removal of water molecule, hence it is also formed by dehydration. This bond is formed between two carbon atoms of two adjacent monosaccharide. So glycosidic bond is formed between two carbon atoms of two adjacent monosaccharides. Duta ওচৰা-ওচৰিকৈ থকা monosaccharide molecular carbon atomৰ মাজত glycosidic bond to form হয় আৰু এই bond to form হওঁতে এটা water molecule remove হয় সেইকাৰণে ইয়াক ডিহাইড্ৰেচনৰ দ্বাৰা ফৰ্ম হোৱা বুলি কয় So protein molecule to form honte keneke dehydration hoi pele eta water molecule release hoy hetu moi dekhwaisu agote so agor class or video to sai loba etu so I have shown how a peptide bond is formed and how a water molecule is released and the process of dehydration I have already explained so check the previous class video Third is ester bond. The ester bond is a bond between the phosphate molecule and hydroxyl group of sugar molecule. So in a nucleic acid, a phosphate moiety or a phosphate links the 3' carbon of one sugar of one nucleotide to 5' carbon of the sugar of the succeeding nucleotide. So that is ester bond. And phosphodiester bond, as there is one such ester bond on either side of the molecule, it is called phosphodiester bond. So what happened? Ester bond is formed between phosphate molecule and hydroxyl group of sugar molecule. So in a nucleotide, a phosphate, then a sugar and nitrogenous bases are formed. So ester bond is formed between the phosphate and the sugar. Okay. But in nucleic acid, DNA, as there is two strands, so there are two strands, and in each strand a ester bond is formed. That's why one such ester bond on either side of the molecule, it is called phosphodiester bond. Phosphodiester, di means two ester. As two ester bonds are found, that's why it is known as phosphodiester bond. So DNA moleculet ki hoy বা নিউক্লিক এসিডছত, মেইনলি ইন DNA, দুটা স্ট্ৰেণ্ড থাকে। এটা স্ট্ৰেণ্ডত থকা নিউক্লিঅ'টাইডবিলাক কানেক্ট হৈ থাকে এষ্টাৰ বণ্ডেদি। কিন্তু যিহেতু ইয়াতে দুটা স্ট্ৰেণ্ড থাকে আৰু সেই দুইটা স্ট্ৰেণ্ডৰ মাজত দুটা এষ্টাৰ বণ্ড ফৰ্ম হয়, সেইকাৰণে ইহঁতক ফস্ফোডাইএষ্টাৰ বণ্ড বুলি কয়। অ'কে, থেংক ইউ।